topic of the masterclass is oxidative stress and the antioxidant pyramid. Monica and we will take you through the oxidative stress and the delicate balance between pro-oxidant and antioxidant molecules to show you how delicate it is, and if not well managed, can easily take to oxidative stress. Focus on the first session will be how, when, why oxidative stress occurs, and how we can fight oxidative stress by using antioxidant solutions, which is the difference amongst the different antioxidant, primary and secondary. And we will try to give you the optimal strategy to reduce and fight oxidative stress in order to improve pet health and well-being. But before to start, as mentioned by Julia, we have prepared a couple of questions. You are not obliged to reply, but would be very kind if you want to give your reply, your answer, because the aim is to understand how much the audience is familiar with the topic. So with antioxidant in general, but also with the differences between primary and secondary antioxidant. So please, Julia, can you launch the first poll question? So, do you know the difference between primary and secondary antioxidant? Yes, no, I never heard about it, or yes, I heard about it, but I'm not sure. So the aim, as I said, is to try to understand how you are familiar with that, because the scope of this masterclass is to let you leave it with a better understanding of the differences between the two of them. Nice. So more than 50% of you are aware of the differences. 36% have heard about them, maybe are not sure. I'm sure that at the end of the masterclass, we will turn the yes into 100%. Thank you, Julia. You can launch the second poll question. What is the most powerful antioxidant in the body? Is it vitamin E? Is it superoxide dismutase? Is it the coenzyme Q10, or is it the vitamin C? Let's see the result. Nice. So I'm not going to give you the right answer now. During my presentation, you will understand, and I will point out the right answer. And so at the end of the session 1, you will be able to give the right answer to that. So thank you very much, Julia. I can start with the first session. The antioxidant demand is increasing in the last years, and that's due to an inclination towards a healthier lifestyle for sure in humans. And as we know, humanization trend is still one of the main trends within the pet food industry, and so such a demand in antioxidant is reflecting also in pet nutrition. The feeling of pet owners is what is good for me is good for my pet. So the way in which I'm supplementing my diet, I want to supplement the diet of my pet. The reason why pet owners supplement their diet and the diet of their pet with antioxidant is usually because they are expecting specific from that. Could be a better physical performance, would be a better control and management of the weight, but they also see antioxidant as a good aid in joint health and stress relief, and indeed they see antioxidant as an anti-aging factors that we will see make a lot of sense. But to understand what antioxidants are and how do they work, we need to have a clear understanding of what reactive oxygen species and free radicals are. The reactive oxygen species are reactive chemical species containing oxygen, and in a biological context, they are natural byproduct of the normal metabolism of oxygen. They are very important for cell signaling and homeostasis, but when in certain situations there is an increase of ROS production, then it could be damaging the cells. Within the family of reactive oxygen species, which I will start calling ROS from now on, free radicals are those molecules with an unpaired electron in the most external layer, which make them unstable, so able to react and to damage other cells. Well, antioxidants are those molecules able either to metabolize reactive oxygen species in less harmful molecules, and that are the primary antioxidant, or to stabilize free radicals by donating them an electron, making them stable, so not able to damage anymore other cells, and those are the secondary antioxidant. So whatever it is the antioxidant molecules, primary or secondary, they are able to protect membrane and cells by the damage which could be caused by ROS and free radicals. In this picture, I want to show you how a normal cell look like and how the same cell would look like under free radicals attack, or on the right side under oxidative stress. And to enable you to visualize it in the real world, take a look at this apple. I'm sure that all of you, once in their life, they have cut an apple, left it on the table, and they have seen that after a few hours, the apple was looking like on the right side of this picture. That's an apple under oxidative stress. And that's the way in which cells and organs of your dog, your cat, of yourself would look like under oxidative stress. But when, how, why oxidative stress occur? The major source of ROS within the cell are the mitochondria. It has been estimated that 90 to 95% of the oxygen passing through the mitochondria is converted into water, while the remaining 5 to 10% is reduced and released in form of free radicals. But there is always the ROS generating system is always coupled with an antioxidant mechanism, which has the aim to maintain the oxidative homeostasis, so to stabilize the ROS that has been produced. But as I said before, there are certain situations in which there is an excessive production of ROS, or that the antioxidant mechanism is not working very well. And so we have an excessive release of free radicals out of the cell. So the free radicals production is a physiological process within the metabolism of oxygen. But beside being a physiological process, there could be an increase in ROS production due to some environmental stressing factors. So due to external factors, which in the case of dog and cats could be an excessive training, vaccination, transportation, very hot season, the aging processes, or other situations such as inflammation, where inflammatory cells, they release a lot, they cause the release of a lot of ROS. Then I have the increased production of ROS, and it means that my animal, so my dog, my cat, he needs to have a very strong antioxidant system in order to fight, to reduce the damage that ROS could cause. If the antioxidant system is strong enough to counteract the reactive oxygen species that is being produced, then I have the maintenance of the oxidative homeostasis. So the balance between pro-oxidant and antioxidant is in favor of antioxidant molecules. On the other side, if I have an increased production of reactive oxygen species, but I do not really prepared with a good, strong antioxidant system, then the balance is in favor of the pro-oxidant molecules, and it leads to oxidative stress. Oxidative stress has multiple effects on the health of the animal. First of all, it causes molecular damages. Molecular damages like denaturation of protein, lipids, nucleic acids, and so on. While at the cellular level, the component that is affected the most by the ROS damages is the membrane. Because of the lipid peroxidation, there is a loss of the structure of the membrane, and so the membrane increase his permeability. The molecular and cellular damages, of course, they reflect on the physiological status of the animal. So the dog, the cat, they will look in a poor healthy status. Their immune system is not going to work efficiently. And there would be an impact, a negative impact on fertility, reproduction, on all the physiological functions. The damage caused by oxidative stress has been recognized as the main feature of several diseases. In the last years, in the human field, scientists have focused on the effect of free radicals and oxidative stress. They looked into the link, and they have established the link between oxidative stress and several diseases, cancer, chronic inflammation, diabetes, several others that you can see here. While in vet medicine, there is the studies are a bit limited, but they're increasing because there is an interest of scientists in understanding how oxidative stress affects pet health. And even more, they are looking into the link between oxidative stress and specific diseases, such as obesity, renal failure, decline on cognitive function, and several others. I choose a few of them to share with you. Of course, I don't have time enough to share all the data that has been published, but I have selected few that I felt will be able to give you the real understanding on how impactful oxidative stress is on pet health and well-being. In humans, oxidative stress has been recognized to be involved in the progression of the chronic renal failure. So what scientists speculated is that the cats under chronic renal failure, they was going to have an high oxidative stress. So what they did, they compared two groups of cats, a group of control healthy cats with a group of cats that were previously diagnosed with chronic renal failure. And they looked in several parameters. I'm going to show you here just two of them, which are quite interesting. First of all, the ratio reduced versus oxidized glutathione, and you see that is significantly higher in the CRF group of cats, which is consistent with the activation of the antioxidant defense mechanism. A second interesting value is the antioxidant capacity, which is strongly and significantly decreased in the CRF cats, which suggests a disequilibrium between the oxidant system and the antioxidant defenses. There were some other data published, but it's clear already from these first results that cats in chronic renal failure, they have an oxidative stress state, and would be important, would be key to restore the oxidative homeostasis to improve their health and well-being. And it's selecting the right pool of antioxidant could be a dietary intervention in restoring the oxidative homeostasis. Obesity is nowadays a big concern in pets, both cats and dogs. It's due to the consumption of a very palatable and nutritious food combined with a very sedentary life. So there is a fat accumulation which result in something like 25–30% of cats being obese, and the percentage is similar in dogs. Obesity is related to several health concerns, but has been also recognized to be linked with oxidative stress and inflammatory, chronic inflammation, inflammatory status. The study I share here on cats, it's an interesting one because scientists they did check and evaluate some, amongst others, inflammatory and oxidative stress parameters in cats that were fed in order to gain weight, get obese, and maintain obesity. They wanted to see how those parameters were changing all over the period while they were the cats were gaining weight. If you see in the table, time 0, 1, and 2, they represent time 0, the optimal weight. So whenever they start the trial, time 1 is whenever the cat get the weight, gain the weight, and get obese, and time is the maintenance of obesity. Looking at the inflammatory markers, you see that the CRP, so an inflammatory protein, which is the C-reactive protein, and the interleukin-6, which is an acute phase inflammatory cytokine, they both are increasing continuously throughout the 3 period, from optimal weight to the maintenance of obesity, which is suggesting a chronic inflammatory response of the cat to the gain weight. As a parameter of oxidative stress has been choose the malonaldehyde, which is a quite common parameter. It gives an understanding of the lipid peroxidation, and you see that the malonaldehyde is as a peak at time 1, so whenever the cat gets obese, but is still high also at time 2, whenever the cat maintain his obesity and accumulate fat. So overall, these results tell us that accumulation of fat lead the cat in a state of oxidative stress, is increasing his oxidative stress parameter, is somehow reducing his quality of life, because his tissues are continuously damaged by an uncontrolled production of oxidant molecules. Obesity is quite a concern also in dogs, and I have highlighted here those few sentences because they are absolutely in line with what we have just seen in cats. The authors, they are here relating the obesity to a decrease in plasma antioxidants. They related obesity also to an increase in oxidative stress markers, and they also related obesity to chronic inflammation, to a chronic inflammatory status. In this case, authors, they did exactly the opposite of what was done in cats. So they did enrich the diet of 12 German Shepherd with a blend of antioxidants. They were mainly secondary antioxidant, and they have looked into the body condition score and in several other parameters that were marker of inflammation and oxidative stress, because they wanted to see how those parameters were changing before and after 6 months, in which there was not a restricted diet, but just a standard diet enriched in antioxidants. Interestingly, what they saw is that the body condition score of the dogs was decreasing. So dogs after 6 months with this diet was in a better shape. The derivatives of reactive oxygen metabolites was significantly decreased. They were half of the before diet. And the biological antioxidant potential was significantly increased. So just looking at these first results, authors were able to conclude that a proper diet with a specific emphasis, giving the right importance to the antioxidant supplementation is important, is key, not just to maintain the animal in a good shape, but also to help maintaining a good oxidative homeostasis. At the beginning of my presentation, I said that antioxidants are seen as an anti-aging factors, and yes, it makes a lot of sense, because as you see from this table, it has been done on a sample of more than 2,000 human beings. You see that the activity of the superoxide dismutase, which is a primary antioxidant enzymes that we will see in a few slides, is decreasing already in the early adult period. It means that after 25, 30 years old, we start decreasing the endogenous production of antioxidants, which are one of the main, or I would say the most important mechanism of defense of our body against oxidative stress. And the same is in dogs. The decline of cognitive function in dogs have been recognized to have a biological basis. The accumulation of damages on lipid and proteins in the brain of senior dogs lead to dysfunction within the neurons. There is, from one side, an increased ROS production, but on the other side, there is a lack in the compensation of the antioxidant from the antioxidant mechanism. So we have this increased production of reactive oxygen species are going to damage several macromolecules within the neuronal cells. Several research have recognized in antioxidants one of the nutraceuticals that would be helpful in this situation, because by reducing the oxidative damage in the brain of the dogs, they were able to slow down the decline in learning and memories. Of course, it's not easy to understand which is the right blend of antioxidant, which is the dosage, long-term effect, when I should start with this dietary intervention, for how long. So several questions that need further investigation, but no doubt that the restoring of the oxidative damage would be helpful. Sorry, the restore of the oxidative homeostasis would be helpful in a senior dog. To understand how to reduce oxidative stress and to fight oxidative stress, we need to start really from the beginning. The major free radicals produced in all physiological and pathophysiological status is the superoxide anion, which needs the superoxide dismutase, which I just mentioned before, so a primary endogenous enzyme, to metabolize it into hydrogen peroxide, which is not a free radical anymore, but still a reactive oxygen species, which need then the other two secondary antioxidant, catalase and glutathione peroxidase, to convert it to detoxify it in not harmful molecules, water and oxygen. These represent the primary antioxidant chain. So the chain of antioxidant working upstream, they are decreasing the free radicals production, working upstream. But whenever the superoxide anion production is too high, or we do not have enough superoxide dismutase to enable his conversion into hydrogen peroxide, you have the production of free radicals. Whenever the free radicals has been produced, you then need the secondary antioxidant chain. So vitamin E, C, A, polyphenols. So all those antioxidants able to stabilize free radicals by donating them an electron. So secondary antioxidant, they work downstream. So the reply to how to reduce oxidative stress, you can do it by working upstream with the primary antioxidant or working downstream with the secondary antioxidant, or even better, you can work on both sides. As I promised you, I would have let you go leave the masterclass with a clear understanding of primary and secondary antioxidant. So I keep go a bit deeper in the differences between the two of them. This is the antioxidant pyramid, a great tool to have a good understanding on how to categorize different antioxidants. At the basis of this pyramid, we have the trace elements and protein, on which I'm not going to spend time because Monica will be explaining them perfectly to you in the second part of this masterclass. I just tell you that they are cofactors, so they are not antioxidants per se, but they are cofactors of the primary antioxidants. Right on top of that, we have the secondary antioxidant. We found them in the feed. They have a buffering role. So we have seen that they are able to stabilize free radicals once they've been already produced, donating them an electron. And the ratio they have is a ratio of 1:1. It means that one secondary antioxidant is able to neutralize one free radical. Those are the glutathione, coenzyme Q10, carotenoids, vitamin E, C, A. Let's say the most common antioxidant we found on the market, also in the pet, used in the pet nutrition. Most of the claim on antioxidant are supported by those secondary antioxidant addition. On the top of the pyramid, you find, first of all, the reply to the question we posed at the beginning of the presentation. So the most powerful antioxidant in the body is the superoxide dismutase. And the superoxide dismutase, together with the catalase and the glutathione peroxidase, they are the three primary antioxidants. They are endogenous enzymes produced by the body. They are essential and they represent a complete defense system. If we look at the ratio, well, the ratio of primary antioxidant is 1 to 1 million. It means that one primary antioxidant is able to metabolize more than 1 million of free radicals. So you can see quite easily the difference between a primary secondary antioxidant in way of working, but also in the way of fighting versus free radicals. We don't have time to go through all of them, each of them, but I just want to give you a few details about secondary and primary to let you understand a bit more their mode of action. Vitamin E is one of the most common widely used vitamins or secondary antioxidant in all the diet. It's we can define it as a chain breaking scavenger. Vitamin E works by stopping the wave of lipid peroxidation. So whenever the wave of lipid peroxidation reaches vitamin E, then vitamin E is oxidized into tocopherol radical and stop the wave and generated by vitamin C, and then there is also the involvement of the glutathione. So you see here there is a longer reaction chain involving several secondary antioxidants. So it's a kind of teamwork amongst secondary antioxidants, which are working by donating electron and stabilizing molecules. Polyphenols is another class, a huge family of more than 8,000 organic molecules. It's another class of secondary antioxidant. Again, they are again very well used and widely used in pet nutrition, but also in human food. And the flavonoids are a subclass of them. It's we can find it in fruit, in vegetable, in cereals, in several ingredients. The way in which they work is again as a secondary antioxidant is donating electron to stabilize free radicals. So they all work downstream. Coming back to the antioxidant pyramid, we have seen how works a secondary antioxidant. We have seen a few examples of them. When it comes to primary antioxidant, first of all, we need to take in mind that they are produced endogenously. And there is always a balance of synthesis and degradation of them. And the superoxide dismutase, I said, is the most powerful antioxidant in the body. And because it's the only enzyme able to transform the most powerful free radical, which is the superoxide anion, into hydrogen peroxide, allowing then the other two primary antioxidants in converting, detoxifying it. So it's really the first, the first enzyme entering in the oxidative pathway is the first enzyme that is avoiding an excess of free radicals and so the oxidative stress to come. Superoxide dismutase can be found in several ingredients. You can find it in algae, pea seed, spinach leaves, tomato leaves, and several other ingredients. But the amount you will find is a tiny amount, and the activity is quite low. In 1989, it has been discovered a special non-GMO variety of cantaloupe melon, which had surprisingly such a long shelf life if compared with the traditional variety of cantaloupe melon. So we were wondering why, and it has been discovered that the reason why was an quite high level of superoxide dismutase. It took around 10 years to optimize and standardize the production of such a variety of cantaloupe melon, but then finally, sorry, nowadays, this variety of cantaloupe melon is the natural vegetable source with the highest content in superoxide dismutase, with an activity which is 20 times higher than the one of the traditional variety of melon. Considering the mechanism of action of the superoxide dismutase, whenever the superoxide dismutase, I said it's a primary enzyme and it's produced endogenously. And whenever it is supplemented as a dietary supplementation, it has been demonstrated to be able to increase the expression of primary enzymes within several organs, adipose tissues, liver, reproductive tract, of the 25 to 30% more, and in several species, which suggests that this is a common process shared by all the aerobic organisms. I shared here just a few examples in lab animals. In rats under cardiac hypertrophy, we have seen that the supplementation with SOD was reducing the cardiac hypertrophy and was showing an increase of endogenous antioxidant within the heart tissue. The same happens with fat hamsters. They had an increase of these endogenous primary antioxidant within the adipose tissue, and they were losing fat tissue. And the same, we were able to show an increase of those primary antioxidant also in the liver of hamsters. But let's do a step further in the mode of action. SOD, we said, is an enzyme. Enzyme is a protein, so it is easily denaturated, and it needs a coating to bypass the stomach in order to avoid the gastric juice to denaturate it and let it lose his beneficial effect. Once the superoxide dismutase has bypassed the stomach, she reached the ileum, the gut, wherever is able to interact with some receptors and stimulate and increase endogenous production of all the three primary antioxidants, which we have seen they are able then to inhibit oxidative stress and its pathway. A couple of years ago, we thought we wanted to show the beneficial effect of SOD supplementation, specifically in dogs, because we were already being able to show it in other species, and we want to make sure that the same effect was going to be also in dogs. So what we were looking was a nice challenge. I mean, for looking the the effect of SOD in fighting oxidative stress, you need oxidative stress to happen. So we need to find a challenge, but we want to respect the well-being of pets. So looking at some scientific review, we thought that maybe using working and racing dogs would have been the right thing, because they would have been the right target, since they are facing a challenge, which is intense physical activity, but it's a good challenge. So we, in collaboration with the vet school of France, Maison Alfort, we investigate the capacity of these superoxide dismutase to reinforce the total antioxidant status of the animal. And we did it taking advantages of a very well-known and famous racing that happens on the Alps in France. And in collaboration with the university, we did a trial starting a couple of weeks, was 13 days before the racing. We involved 18 Alaskan Huskies, adult between 3 to 6 years old. The supplementation with the SOD started 13 days before the first race. We took 3 samples of blood. The first sample was the day before the first race. Then the dog was racing 3 days in a row. Then we got a second blood sampling the day after the third race, and 1 week after, to see how the dog were recovering. What we look in these samples was as a parameter, we look at the total antioxidant status, which reflects the antioxidant capacity of the dog in a global way. It means that it includes both enzymatic and non-enzymatic antioxidants. In the results, we saw that the total antioxidant status, you see the green bars are the melon sod supplemented dogs, and you see that the green bar are quite stable during the whole period. It means that the antioxidant capacity of the animal was supplemented with SOD was stable throughout the period. But even better, right after the third race, the antioxidant status of the melon sod group was significantly better than the control group. And at T3, whenever it was like 1 week after the stop of the racing, we don't have a significance here, but it's not surprising because the dogs used are all very healthy and well-trained dogs. So we would expect all of them to recover well 1 week after the last race. So we were able to conclude that a melon sod supplementation is able to maintain the antioxidant status of the dog during intense physical activity. But I would add that here the challenge, it's not just a physical challenge, but there is also a mental challenge because there is a very high competition during those races. So it was also a kind of mental stress for the dog. So we were able to reinforce the antioxidant defense of the dog. Equine are another species of the animal which are exposed to inflammation and oxidative stress. And in this case, we did a trial in which we were able to collect samples, not just from the blood, but also from the synovial, from the joint. So we took some sample of synovial fluid. We looked into several parameters, but I choose just a few of them here to show to you. Then you are more than welcome to ask if you have interest, and we will be more than happy to share additional details. In this trial was a 23 days trials in which we involved 2 groups of 8 horses, each of them, a control group versus a group of 8 horses that were supplemented with the melon sod. They got the supplementation for 22 days, and after the 22 days of supplementation, they got a very intense training. And we did blood and the synovial fluid sampling 1 day before, 1 hour after, and 1 day after this intense training. And here below I show just some of the results. Quite interesting. You can see that the total antioxidant status in the synovial fluid is stable for the orange bar, while for the control there is a drop right after 1 hour after the training, while the supplemented horses, they stay very stable with their antioxidant capacity. The superoxide dismutase show a significant increase in the supplemented horses. So they were increasing the production of primary antioxidant. And a very good one is the results we got on the glycosaminoglycan. Glycosaminoglycan is a very important parameter because it is an indicator of the cartilage catabolism. So I want this value to be always low, because if it's low, it means that my cartilage is still elastic. So you see here that there is also for the glycosaminoglycan, there is a stable value for the melon sod group, while for the control group, you have a peak of glycosaminoglycan, which means catabolism of the cartilage. So the cartilage was losing their elasticity, 1 hour after the intense training. So thanks to these results and some others, we were able to say that the supplementation with the melon sod was able to stimulate antioxidant defense in horses and to maintain the integrity of their cartilage. Well, I can conclude my session. Just the key message I would like you to take home are that the free radicals production is a physiological process, as we have seen within the metabolism of oxygen. And usually there is an antioxidant system able to maintain the oxidative homeostasis. But there are certain situations in which you have an increase of ROS release, or you have a less availability of antioxidant, and then oxidative stress can occur. Oxidative stress is is involved in the aging process, can occur also after very intense physical activity, but it is also linked to several diseases such as cancer we have seen, or some obesity, or some others. So it's important to reduce oxidative stress, to maintain the oxidative homeostasis, and you can do these by using secondary antioxidant to neutralize free radicals, or you can use the primary antioxidant working upstream, controlling so the free radicals production. But we truly believe that the best antioxidant strategy should combine primary and secondary antioxidant in order to target different steps within the oxidative pathway and obtaining in this way a synergistic effect. So I thank you very much for your attention. And if you have any question, I would be more than happy to reply.